This frog is not bleeding out, it is hiding its blood. During the day, a northern glass frog can sleep beneath a leaf with its body flattened against the green surface. Light passes through its skin and muscles, its outline fades. Even the red network of blood vessels seems to disappear. The blood is still there, but most of its red blood cells have been pulled out of circulation and packed inside its liver. By hiding the most colorful part of its blood, the frog becomes dramatically more transparent. That sounds impossible for a living vertebrate. Blood must move, oxygen must reach the tissues, and cramming blood cells into one organ sounds like an excellent way to cause a deadly blockage. Yet this tiny frog appears to do it every day. Glass frogs live in humid forests of Central and South America, often near streams. They are mainly active at night. That is when they move across vegetation, hunt, call, and breed. When daylight arrives, some species rest upside down beneath broad leaves. This creates a problem. A motionless frog is easier to inspect, and a leaf can become a bright green panel when sunlight passes through it. Anything sitting on that leaf may cast a recognizable frog-shaped shadow. Ordinary green camouflage helps, but it is not enough. A frog painted the perfect shade of leaf green would still have a sharp edge. A searching predator could detect the boundary between frog and background. Glass frogs weaken that boundary. Their name comes from the transparent or partly transparent skin on the underside of the body. Viewed from below, the bones and internal organs can be visible, but calling them completely transparent is misleading. Their backs still contain green pigment, and several internal structures remain opaque. They are closer to tinted, living glass. That imperfect transparency turns out to be useful. When light travels through the frog, its apparent brightness changes with the leaf beneath it. On a brighter background, the frog appears brighter. On a darker background, it becomes darker. It does not perfectly copy every vein and spot on the leaf. Instead, it passively adjusts to the general brightness of its surroundings. The effect is strongest around the legs. A resting glass frog tucks its limbs tightly beside its body, and those limbs can be more translucent than the center. Instead of an abrupt jump from leaf to frog, there is a gradual transition. Leaf, translucent leg, greener body. Researchers call this edge diffusion. The frog does not need to change color like a chameleon. Light passing through its tissues softens its silhouette. In a 2020 study, researchers tested this idea using visual models, human detection experiments, and translucent gelatin frogs placed on leaves in Ecuador. The translucent models were attacked less often than opaque versions. The camouflage did not make the frogs literally invisible. It made their outlines less obvious, which can be enough to delay detection. But there was still a glaring obstacle to transparency: blood. Most vertebrate tissues are difficult to make transparent because they contain many surfaces and structures that scatter light. Blood adds another problem. Red blood cells are filled with hemoglobin, the molecule that carries oxygen. Hemoglobin strongly absorbs parts of visible light, including wavelengths that matter against a bright green leaf. Imagine building a clear house and then running dark red pipes through every wall. The walls might transmit light, but the plumbing would reveal the entire structure. A living frog cannot simply stop circulating blood. Its tissues still need oxygen, and carbon dioxide and other wastes must still be moved. Yet sleeping northern glass frogs seem to come remarkably close to clearing the red cells out of the visible parts of their bodies. The clue appeared when researchers compared sleeping frogs with active ones. While resting, the frogs looked clearer. When they woke, moved, or were stressed, red blood cells rapidly returned to vessels in the skin and muscles. The frogs became more opaque. Studying this was unusually difficult, because normal research procedures could destroy the very phenomenon being measured. Handling, anesthesia, or stress could wake the frog and send its blood cells back into circulation. The scientists needed to look inside a comfortably sleeping animal without cutting it open or injecting a visible dye. They used photoacoustic imaging. The basic idea is surprisingly simple. A carefully controlled pulse of light enters the body. Molecules that absorb that light warm by a tiny amount and generate ultrasonic waves. Sensors detect those waves and use them to map where the absorption happened. Because hemoglobin is an excellent light absorber, the system can reveal red blood cells hidden inside tissues. It effectively listens for the locations where blood absorbs light. The resulting maps showed that, during sleep, the red blood cells were not vanishing. They were gathering inside the liver. In the studied species, Hyalinobatrachium fleischmanni, red blood cell signals in the skin and muscle fell by an average of about 89% during rest. The liver's large internal channels, called sinusoids, expanded and filled with densely packed cells. The organ increased in volume by roughly 40%. At the same time, the frog's transparency increased 2 to 3-fold. The liver acted like a biological storage vault, accepting most of the red cells that would otherwise trace dark vessels through the frog's body. Even the vault is disguised. The liver and several other organs are enclosed by tissue containing reflective guanine crystals. These crystals form a bright, mirror-like covering that blocks much of the visible light entering the organ. The frog does not just move its red cells into one place. It moves them behind a reflective screen. Then, night returns. The frog wakes, metabolic demand rises, and the stored red cells flow back into circulation. Hemoglobin carrying oxygen once again spreads through the vessels. After exercise in the study, the frogs gradually recovered their resting level of transparency over about an hour as red cells reaccumulated in the liver. This reveals the trade-off behind the trick. Maximum transparency is best suited to a motionless animal with low energy demands. A frog actively jumping, calling, or hunting needs the oxygen-carrying capacity of those cells more urgently than it needs extreme transparency. During the clearest resting state, only a very small fraction of the frog's total hemoglobin was carrying oxygen in circulation. Researchers think the frogs may sharply lower their metabolism during sleep, rely partly on metabolic pathways that do not require oxygen, or use some combination of physiological strategies. Exactly how they tolerate the oxygen limitation remains unresolved. And then there is the clotting mystery. A blood clot is not simply a pile of red cells. It is a controlled response involving blood cells, clotting proteins, and a stabilizing mesh that seals damaged vessels. But slow flow and extreme cellular crowding can create dangerous conditions. In most vertebrates, a dense concentration of blood cells inside a vessel would raise serious concerns about blockage and tissue damage. The glass frog repeatedly packs somewhere around 82 to 93% of its red blood cells into its liver, then releases them again, apparently without triggering catastrophic clots. Scientists do not yet know how. The answer could involve unusual clotting chemistry. The red cells might change shape or arrangement while packed. The liver's sinusoids may have special physical properties. The frog could also use multiple safeguards at once. Until those mechanisms are identified, claims that this animal has already provided a treatment for human blood clots go far beyond the evidence. What it does provide is a living example of safe, reversible blood cell storage. Because frogs share important components of blood clot regulation with mammals, understanding this system may eventually reveal useful principles. But first, researchers have to learn what prevents the storage vault from becoming a trap. There is another important limit. The blood-hiding discovery came primarily from one northern glass frog species. Glass frogs vary greatly in pigmentation, organ visibility, and transparency. We should not assume every species uses the same mechanism to the same degree. Transparency itself is also fragile. Research published in April 2026 found that translucent frog models matched leaf brightness more closely, but conspicuous opaque markings could erase that camouflage advantage. If bones, organs, or patterns create recognizable high-contrast shapes, making the surrounding tissue clearer may actually expose them. That may help explain why a glass frog is not simply a perfectly clear frog. Green pigment on the back can conceal internal structures, while the clearer belly and limbs allow light to soften the animal's boundary. Its camouflage is a compromise between showing the background and hiding what is inside. So the sleeping frog beneath the leaf is not invisible. Look closely, and you may see its eyes, bones, organs, and a small mirrored patch where its blood has been stored. But a predator does not examine every leaf in a laboratory. It scans a crowded rainforest filled with moving shadows, veins, water droplets, and shifting light. Survival may depend on being overlooked for one more second. That is what the glass frog has engineered with its own body. Transparent skin reduces the barrier. Translucent legs erase the edge. Green pigment covers the structures that cannot disappear. And when the frog becomes still, its blood retreats into a reflective liver. The strangest part is not that this frog has almost nothing to hide. It is that becoming transparent requires it to hide nearly everything in exactly the right place.